Hello friends, this is Mr. Vipul Sonsare. Once again welcome you in our new video lecture. Today we are going to see important identification test for the crude drugs. So different chemical tests which are generally used for the identification of the various crude drugs that we will see in today's video lecture. So we will see in the form of tabular form that will be better for understanding. So first of all we will see the test, that is Vitalimorine test. So this Vitalimorine test uses the concentrated nitric acid and methanolic potassium hydroxide as a reagent, and it is important in the identification of tropane alkaloids. The tropane alkaloids which are present in belladonna, hyoscyamus, these tropane alkaloids are identified using Vitalimorine test, and these tropane alkaloids shows the violet color in presence of this reagent. Second test which is used for the identification of cinchona alkaloids. So the test is phthaloequine test, which use the ammonia solution and bromine water, and the cinchona alkaloids shows the green color in presence of the reagent. So the cinchona alkaloids which are present in the cinchona bark are basically identified using phthaloequine test. Next, the ball-trigger test. It is used for the identification of anthraquinone glycosides in senna. So in ball-trigger test, the dilute sulfuric acid, chloroform and ammonia are used as a reagent, and this anthraquinone glycosides shows the pink red color. Next, the Murexide test. This Murexide test is specifically used for the identification of purine and xanthine alkaloids. For example, caffeine. The caffeine contain purine alkaloids. So in Murexide test, the hydrochloric acid and potassium chlorate is used as a reagent, and the purine alkaloid showed the purple color in presence of the reagent. Then just see the test for identification of digitalis, which contains the cardiac glycosides. So first test is a Baljet test, which use the sodium picrate as a reagent, and this digitalis shows the yellow or orange color. The second test, that is legal test, which use the sodium nitroprusside and sodium hydroxide and pyridine as a reagent, and this shows the pink or red color. These both tests are used for the identification of digitalis, which contains the cardiac glycosides. Next, that is borax test. We use the borax as a reagent, and it is used for the identification of aloe, which contains the glycosides. So this aloe shows the green fluorescence in presence of borax. Next test used for the identification of all alkaloids, that is ammonium rennicate test. So it used the ammonium rennicate and hydroxylamine as a reagent, and this alkaloid shows the pink precipitate in presence of the reagent. The next test, that is picoulonic acid test. This test is also used for the identification of alkaloids. It used the picoulonic acid as a reagent, and alkaloid shows the yellow pipette in presence of picoulonic acid. The next test, tannic acid test. It is again used for the identification of alkaloids, and use the tannic acid solution as a reagent, and this alkaloid shows the buff color precipitate in presence of tannic acid solution. Just see next the Halfin test, which is used for the identification of cholesterol which is present in the wool fat. So in Halfin test, acetic anhydride and sulfuric acid is used as a reagent, and this cholesterol in wool fat shows the deep green color in presence of the reagent. Just see the test for the identification of steroids. So first is Salkowski test, which use the chloroform and concentrated sulfuric acid as a reagent, and the steroids, in presence of steroids, the acid layer shows the greenish yellow fluorescence that indicate the presence of steroids. The second test, that is Lieberman reaction. So it used the acetic anhydride and concentrated sulfuric acid, and the steroid shows the blue color. So the steroids are identified using two tests, that is Salkowski reaction and Lieberman reaction. Just see the test for the identification of ipecac, that is Epicocuna. So biological source, that is Syphilis epicocuna, is identified using Froday's test, which use the sodium molybdate and sulfuric acid as a reagent, and this ipecac shows the violet color in presence of reagent. Next, see the Cades test, which use 90% 3,5-dinitrobenzoic acid, and it is used for the identification of unsaturated lactone ring in cardiac glycosides. The digitalis glycosides, right, contains the unsaturated lactone ring attached to the steroidal nucleus, and this lactone ring is identified using Cades test, which shows the purple color in presence of reagent. Next, the umbelliferone test. This umbelliferone test, as such name suggests, it is used for the identification of umbelliferone in Asafoetida. So it used the concentrated hydrochloric acid and ammonia as a reagent, and this umbelliferone shows the blue fluorescence in presence of the reagent. Next, the Kahler-Killani test. This Kahler-Killani test used for the identification of digitoxose, that is sugar, which is present in digitalis glycosides. So glycosides contain two different parts, that is sugar and non-sugar. So the sugar in a digitalis glycosides is identified using the Kahler-Killani test. In Kahler-Killani test, the lead acetate, chloroform and ferric chloride is used as a reagent, and which shows the reddish brown to bluish green color in presence of digitoxose sugar. Next, the Lieberman-Burkhardt reaction or Lieberman-Burkhardt test. It used for the identification of sterols and triterpenes as well as bufadienolides. Bufadienolides, that is your glycosides. So chloroform and acetic anhydride and sulfuric acid is used as a reagent, and the sterols and triterpenoids shows the green color in presence of the reagent, that is Lieberman-Burkhardt test. Next, the test for identification of isobarbaloin, the specific constituent in aloe. So the Claus test or caprolone test is used for the identification of isobarbaloin in aloe, which use the copper sulfate, sodium chloride and alcohol as a reagent, and this isobarbaloin show the red color, right, that is Kurkau aloe. The isobarbaloin in Kurkau aloe shows the red color. Then next test, that is Raymond test. This Raymond test is used for the identification of cardiac glycosides, right, like digitalis glycosides. So it used the hot methanolic alkali as a reagent, and this cardiac glycoside shows the violet color in presence of reagent. Next, the two important tests for the identification of saponin glycosides. So first is foam test. So the saponin glycosides have property, that is that these glycosides produce the foam in presence of water. So it can be identified using foam test. And second test, the hemolytic test. So when the extract of saponin glycosides treated with 1 drop of blood, the clear hemolytic zone is appear. That indicates the presence of saponin glycosides. Next test is used for the identification of cyanogenic glycosides, which are important component of bitter almond or wild cherry. So these cyanogenic glycosides are identified using Gugnard reaction or sodium picrate test. So when the moistened drug is placed in contact with sodium picrate paper, the sodium picrate paper turn the brick red or maroon color. The yellow color sodium picrate turn to the brick red or maroon color paper. That indicates the presence of cyanogenic glycosides. So just remember the name of test. Next, two important tests for the identification of flavonoids. So first is Shinoda test, which use 90% ethanol, then concentrated hydrochloric acid and magnesium. So in presence of this reagent, the flavonoid shows the orange or red to purple color. The second test for the identification of flavonoids, that is sulfuric acid test, which use 80% of sulfuric acid as a reagent, and this flavonoid shows the deep yellow color. Next, that is test for the identification of tannins. So most popular test, that is gold beater skin test. So in this test the gold beater skin is used. So this gold beater skin, when treated with 25% hydrochloric acid and then 15% ferrous sulfate, the brown or black color is produced on the skin. So brown or black color skin indicate the presence of tannins. So in this identification test, what you will remember, just remember the name of test and the drug which is identified, along with the most important thing, that is reagent, because in GPAT the question may ask on the reagent as well as the drug also, right. Just see the important question which has appeared in GPAT 2009, that is Clon's test is used for the identification of. So this Clon's test, or also known as Caprilloin test, is used for the identification of isobarbaloin in aloe. The important answer, that is correct answer, is isobarbaloin. Next, gold beater skin test is used to detect the presence of which of the following class of compound. So this question has appeared in GPAT 2011. So as we have discussed earlier, the gold beater skin test is used for the identification of tannins. So correct answer, that is option A, tannins. Next, leaves of digitalis purpurea gives positive. So the test, name of test is basically asked which of the following test where the digitalis purpurea leaves gives the positive test. So this question has appeared in GATE 2003. So of course the correct answer is legal test, right, because this bond tagger test is used for identification of anthraquinone, Murexide test used for identification of purine alkaloids, and Thioequine test is used for identification of cinchoalkaloids. So remaining answer, that is option B, that is legal test. So these are some important identification tests of crude drugs. Thank you.